हेलो एवरीवन, कैसे हैं बच्चों आप सब लोग। एंड आई होप कि आप सभी लोग बहुत बढ़िया होंगे एंड अपनी तैयारी जैसे जैसे आपके नीट के दिन आ रहे हैं पास में, आप बहुत तेज़ी के साथ में स्पीड बढ़ा करके रिवीजन अपना कर पा रहे होंगे। और ये पॉडकास्ट जो मैं आप लोगों के लिए प्रिपेयर कर रही हूँ, एंड डेफिनेटली ये पॉडकास्ट आपकी हेल्प कर रहे होंगे। मुझे कॉमेंट सेक्शन में अपने व्यूज़ ज़रूर शेयर करिएगा। एंड आज हम करने वाले हैं प्रिंसिपल्स ऑफ इनहेरिटेंस एंड वेरिएशन। बट उससे पहले अगर आप मेरे चैनल पर नए हैं तो बेटा सब्सक्राइब कर लीजिए, और अगर आपको लगता है कि ये वाकई हेल्पफुल है ना तो इसे दोस्तों के साथ में शेयर ज़रूर करिएगा एंड ऑल्सो प्रेस द लाइक बटन। चलिए स्टार्ट करते हैं प्रिंसिपल्स ऑफ इनहेरिटेंस एंड वेरिएशन को। इसमें सबसे पहले हम देखने वाले हैं इंट्रोडक्शन। सो जेनेटिक डील्स विद इनहेरिटेंस एंड वेरिएशन ऑफ कैरेक्टर्स फ्रॉम पेरेंट्स टू ऑफस्प्रिंग. Inheritance is the process by which characters are passed on from parent to progeny. Variation is the degree by which progeny differ from their parents, and human knew that the causes of variation was hidden in sexual reproduction. Next आता है Mendel's laws of inheritance. So Gregor Mendel conducted hybridization experiments on garden peas for 7 years from year 1856 to 1863. And proposed the law of inheritance. Mendel selected 14 true breeding pea plant varieties as pairs, which were similar except for one character with contrasting traits. And the contrasting traits and character which he has taken are stem height, tall and dwarf, flower color, violet and white, flower position, axial and terminal, pod shape, inflated and constricted, pod color, green and yellow, and seed shape, round or wrinkled, and seed color, yellow or green. Next है inheritance of one gene. Mendel found that F1 always resembled either of the parent, but in F2 both traits appeared. 3/4 showed the dominant trait and 1/4 showed recessive. Both traits were identical to their parental type and did not show any blending. That is, none were intermediate type. Mendel got similar results for all traits. To determine the genotype of dominant trait at F2, Mendel performed test cross. Based on monohybrid cross, Mendel proposed two general laws. Law of dominance explains the expression of only one parental character in F1 of monohybrid cross and both F2. It also explains the proportion of 3:1 obtained at the F2. Law of segregation. The factors or alleles of a pair segregate from each other such that gametes receive only one of the two factors. Incomplete dominance. F1 did not resemble either of the parents and was in between the two, seen in dog flower, snapdragon or antirrhinum species. In this case, capital R has red flower, small r has white flowers, but capital R small r has pink flowers. Here genotypic ratio at F2 is like Mendelian monohybrid cross 1:2, 1:1, but phenotypic ratio changed from 3:1 to 1:2:1. Now the explanation of concept of dominance. In diploid organism, there are two copies of each gene, that is a pair of alleles. One of them may be different, that is modified. The normal allele produces normal enzyme needed for transformation of substrate. Number 2, if the modified allele produces normal or less efficient enzyme which produces same phenotype or trait, it is dominant. But if it produces non-functional or no enzyme, the phenotype will be affected and recessive trait is seen. Factors or genes are the units of inheritance. They contain information required to express a particular trait in an organism. British geneticist R.C. Punnett developed a graphical representation called Punnett square to calculate possibility of all possible genotype of offsprings in a genetic cross. Codominance. F1 resembles both parents. ABO blood group in human being is controlled by gene I and having three alleles IA, IB and small I. IA and IB produces slightly different form of sugar while small I does not produce any sugar. IA and IB are completely dominant over small I, but when IA and IB are present together, they express their own sugars. And because of codominance, hence the BC have both sugars. There are 6 genotype and 4 phenotypes in human ABO blood types. Next is pleiotropy. A single gene can exhibit multiple phenotypic expression. It is the effect of a gene on metabolic pathways which contribute towards different phenotypes. Examples: number 1, phenylketonuria, single gene, and it causes mental retardation and reduction in hair and skin pigmentation. And number 2 is a starch synthesis gene in pea, and gene is having two expressions: the seed shape, that is the dominance, and size of starch grain, that is incomplete dominance. Now the capital B large grains and capital B intermediate size grain, but the shape is round, and small b small grains and the shape is wrinkled. Now multiple alleles. More than two alleles governing the same character. ABO blood grouping is a very good example of multiple alleles. Since in an individual only two alleles are present, multiple alleles can be found only in population studies. Inheritance of two genes. Mendel also worked and crossed pea plants that differed into two characters and got a phenotypic ratio of 9:3:3:1 and genotype ratio of 1:2:2:4:1:2:1:2:1. Based on these dihybrid crosses, Mendel proposed a law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. Next is chromosomal theory of inheritance. Walter Sotten and Theodore Boveri noted that the behavior of chromosomes was parallel to behavior of genes, and they used chromosomes movement to explain Mendel's law. Sotten united the knowledge of chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. Experimental verification was done by T.H. Morgan, who worked with fruit flies Drosophila melanogaster. Next is Drosophila melanogaster were suitable for genetic studies. So characteristics: they could be grown on simple synthetic medium in laboratory. They complete their life cycle in about 2 weeks. A single mating could produce a large number of progeny flies. There is a clear differentiation of sexes. Also, it has many types of hereditary variations that can be seen with low power microscopes. Now the next is linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked, similar to the dihybrid crosses of Mendel in peas. But the F2 ratios deviated significantly from 9:3:3:1 that is expected when the two genes are independent. When two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. Morgan attributes it to physical association or linkage of two genes and used term recombination to describe non-parental gene combinations. Some genes were very tightly linked, showed very low recombinations, while others were loosely linked and showed higher recombinations. Morgan student Alfred Sturtevant used frequency of recombination between genes on the same chromosome as a measure of distance between genes and mapped their position on chromosome. Today, genetic maps are extensively used as a starting point in sequencing whole genomes, as done in case of Human Genome Project. Next is polygenic inheritance. Traits controlled by 3 or more genes are polygenic traits. It also takes into account influence of environment. The phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. Example: human skin color. Capital A, capital A, capital B, capital B, capital C has darkest skin color. Small a, small b, small b, small c has lightest, and the capital A, small a, capital B, small b, capital C, small c intermediate color. Other example is height in humans. Sex determination, genetic or chromosomal basis. Initial clue came from insects. The X body of Hanking was in fact X chromosome. Types: XO type, male heterogamity, example grasshopper. XY type, male heterogamity, example insect, man. ZW type, female heterogamity, example birds. Now sex determination in humans. Genetic makeup of sperm determines sex of the child, and in each pregnancy there is always 50% probability of a male or female child. It is unfortunate that in our society females are blamed for giving birth to female children. Now the sex determination in honeybee. Haplodiploid sex determination: an unfertilized egg develops as male, it means drone, that is haploid, queen and worker bees females are diploid. Next is mutation. Results in alternation of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. Loss or gain of a segment of DNA results in chromosome alteration. Alteration in chromosome results in abnormalities or aberration. Chromosomal aberrations are commonly observed in cancer cells. Mutation also arises due to change in a single base pair of DNA known as point mutation, example sickle cell anemia. Deletions and insertions of base pairs of DNA causes frame shift mutation, and chemical and physical factors that induce mutations are called mutagens. Example, UV radiations can cause mutations in organisms. Next is genetic disorder. Pedigree analysis. Controlled crosses are not possible in case of human beings. Study of family history about inheritance of a particular trait provides an alternative. Such analysis in several generations of a family is called pedigree analysis. Now the symbols used in the human pedigree analysis, this you have to see from your NCERT, the symbol, and that is for the male, female, sex unspecified, affected individuals, mating between relatives, and parents above and children below, and parents with male child affected with disease, and 5 unaffected offspring. These symbols you have to see from your NCERT. Now the Mendelian disorders, mainly determined by alteration or mutation in a single gene. It may be dominant or recessive, autosomal or sex-linked. Example, color blindness, that is sex-linked recessive and due to defect in either red or green cone of eye due to mutation in certain genes present on X chromosome. 8% of males and only about 0.4% of females affected. Next is hemophilia. X-linked recessive single protein, that is a part of cascade of protein involved in blood clotting is affected. Then after sickle cell anemia, autosome linked recessive controlled by a single pair of allele HbA and HbS. Next is phenylketonuria, inborn error in metabolism, autosomal recessive, affected individual lack enzyme which converts phenylalanine to tyrosine and results in mental retardation. Then after thalassemia, autosomal recessive could be due to mutation or deletion. Alpha thalassemia controlled by two closely linked gene HbA1 and HbA2 on chromosome 16, and beta thalassemia controlled by single gene HBB on chromosome 11. Then after chromosomal disorders caused due to absence or excess or abnormal arrangement of one or more chromosomes, failure of segregation of chromatids during cell division cycle resulting in gain or loss of a chromosomes is called aneuploidy, and failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism. This is called polyploidy, often seen in plants. Trisomy or monosomy leads to very serious consequences in the individual. Common examples of chromosomal disorders, number 1 is Down syndrome. Trisomy of 21 was first described by Langdon Down in 1866, and the symptoms are short-statured, small round head, furrowed tongue, partially open mouth, palm is broad with palm crease, and physical psychometer and mental development is retarded. Klinefelter syndrome, karyotype 47XXY, overall masculine development. However, gynecomastia is also expressed. Such individuals are sterile. Next is Turner syndrome, due to absence of one of the X chromosome, that is 45 with XO. Such females are sterile as ovaries are rudimentary besides lack of other secondary sexual characters. Now the pedigree analysis for the autosomal dominant trait, example है myotonic dystrophy, and autosomal recessive trait, example है sickle cell anemia. T.H. Morgan found that the Drosophila, the genes for yellow body and white eye, were very tightly linked and showed only 1.3% recombination, while white eye and miniature wing showed 37.2% recombination. In honeybee, males produce sperms by mitosis. They do not have father and thus cannot have sons, but have grandfathers and can have grandsons. Cystic fibrosis is autosomal recessive disorder. Chromosomal disorders can be easily studied by the analysis of karyotypes. Inheritable mutations can be studied by generating a pedigree of a family. and the family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. और इसी के साथ ही आपका ये चैप्टर कंप्लीट हो गया है एंड आई होप कि आपको इसका शॉर्ट नोट्स का ये पॉडकास्ट जिसमें सारी एनसीईआरटी की इंपॉर्टेंट लाइन मैंने आप लोगों के लिए पढ़ दी हैं आप लोग के रिवीजन में ज़रूर हेल्प करेगी. सो इफ़ यू आर न्यू ऑन माई चैनल देन डू सब्सक्राइब एंड प्रेस द लाइक बटन एंड ऑल्सो शेयर विद योर फ्रेंड्स सो दैट इट कैन हेल्प ईच एंड एवरी नीट एस्पिरेंट्स. So see you soon in the next podcast. Till then you can share your views in comment section with me so that I can implement them from the next podcast. So see you soon in the next one. Till then just stay tuned with me. Bye bye. Take care.